Welcome. My name is Ivan Rich and I'm the CEO of Hemogenix, your outsourcing partner for in vitro contract research services. I shall be your tour guide for the next few minutes. Hemogenix started in 2000 and is now 18 years old. A teenager compared to other companies, but a company that prides itself on performing high-quality science. In 2003, Hemogenix relocated from Columbia, South Carolina to Colorado Springs, Colorado. After relocating the company, we started developing our patented and proprietary assays into assay kits for in-house use. Hemogenix divested the assay kit business in 2017 to Preferred Cell Systems. Hemogenix is a privately owned company that works with small, medium, and half of the top 50 biopharmaceutical companies in the world. We may be a small company, but we have made some big, notable achievements and milestones in the field of in vitro toxicology, basic research, and cellular therapy. The company's name, Hemogenix, implies that we focus on the blood-forming or hematopoietic system, as well as the immune system. However, Hemogenix has since developed its own proprietary assays for several other types of in vitro toxicity testing. The importance of this will become apparent in a few moments. Indeed, our in vitro toxicity testing platforms are used by many biological systems and cell types, including ES and iPS-derived cells, transformed cell lines, and tumor cells, and can usually be obtained from multiple species. Much of our patented or proprietary technology incorporates standardized and validated bioluminescent readouts that provide the greatest sensitivity and accuracy, especially for rare populations such as primary stem cells. Historically, in vitro hematotoxicity testing has been used to study the response of drugs and other compounds for their neutropenic, thrombocytopenic, and/or anemic effects. This has generally been done using the traditional methylcellulose colony-forming cell, or CFC assay. However, due to its insensitivity and inaccuracy and the use of viscous methylcellulose, the CFC assay cannot be used for high-throughput screening or to detect responses of the rare and primitive stem cells. Since the stem cells are usually more sensitive to perturbations, using the CFC assay misses the most important predictive information for hematotoxicity testing. With the introduction of the Halotox HT platform, the deficiencies inherent in the CFC assay have been completely obviated. Halotox HT is capable of detecting up to 9 different stem cell populations and 13 progenitor cell populations, including multiple T and B lymphopoietic cell populations from up to 9 species. With 96 or 384 well high throughput capability and demonstrating a high degree of in vitro to in vivo concordance, Halotox HT is one of the most advanced in vitro toxicity platforms available. Halotox HT has been validated according to FDA bioanalytical method validation guidelines and against the Registry of Cytotoxicity Prediction Model. Using different culture methods but the same readout, Hemogenix developed Immunoglotox HT for immunotoxicity studies. Primitive lymphopoietic cells are detected using Halo, but different populations of T and B cells are measured using the Immunoglotox HT platform. Recent developments have allowed Hemogenix to develop Immunoglotox TCP for different lymphocyte populations and Immunoglotox BCP for B lymphocyte populations. We can even use our assays to work with myeloid-derived suppressor cells, or MDSCs. Hemogenix is one of the few companies that has these capabilities. Although Hemogenix is known as the leader in hematotoxicity testing, it has developed in vitro toxicity assays using the same readout to measure hepatotoxicity, cardiotoxicity, renal toxicity, neurotoxicity, skin toxicity. By combining these with other assays for hematotoxicity and immunotoxicity, the very powerful Comparatox platform is formed. Comparatox is one of the few in vitro toxicity testing platforms that uses exactly the same readout for all 7 types of toxicity testing. As a result, you can directly compare and rank any number of drug candidates or xenobiotics according to their response and/or toxicity in different tissues and different species. Some of these advanced in vitro toxicity assays have also been incorporated into a panel of assays used to characterize in vitro toxicity of nanoparticles. The Nanotoxicity Characterization Laboratory of the National Cancer Institute have recommended basic assays. Our advanced methodology can be used as alternatives to these recommended by the NCL and elevate nanoparticle toxicity testing to yet another level. All of the toxicity platforms described have been developed with multiplexing in mind. This allows mechanism of action studies to be performed, usually on the same sample being tested, for potential toxicity. This saves time and money, 2 aspects that Hemogenix is acutely attuned to. Innovation is a Hemogenix trait. Besides developing assays to analyze cellular drug-drug interaction and change in cellular response due to repeated drug dosing, Hemogenix was the first company to develop specific assays for different steps in the cellular therapy process. From testing stem cell quality and potency to monitoring engraftment and reconstitution in patients after stem cell transplantation. Finally, what is our workflow to help you achieve the goal you want to outsource? Many companies contact us when they see unexpected results from preclinical animal studies. We can help you much earlier in the drug development pipeline to save time and money using our screening technologies that focus on primary cells and are highly predictive of the human response. However, we also work with CROs to perform ex vivo, in vitro studies of organs and tissues. These gain a far greater insight into the drug response that traditional toxicity studies cannot offer. To help us suggest which methods will help answer your goals, we may need to discuss things in more detail, and are happy to sign a CDA or NDA to achieve this. We will try and provide the most cost-effective route to achieve your goals using the most advanced assays at our disposal. We will prepare a detailed study plan for your approval. Studies can be performed under GLP or non-GLP conditions. Many of the studies we perform are included in INDs. Most studies are completed within 4 to 7 days, depending on the assays used. This means that we can provide a draft report within 5 to 7 business days after study completion, and the final study report will include all of the raw data. Thank you for taking the time to view this video. We hope we can be of service to you in the near future.